Hello, folks. Welcome. Everybody's pouring in. We'll give you a couple of minutes before we get rolling here. But we're glad to see you. I think we're going to have a pretty amazing crowd today. Welcome, everybody. We'll just give you a couple of more minutes and we'll get rolling. And I'm going to give people one more minute to get in the door. All right. Again, welcome to everyone. Probably be some others showing up, but we're going to get rolling here because we got an exciting show. I'm Dave Sly, Conservation Director for Wild Virginia, and we're thrilled to have you here. I'm going to give you a little bit of introduction, but not much because I want you to spend most of this listening to Alissa and learning. I will tell you that I've been excited about this for quite some time because I love mussels. And if you don't love them, you will, I think, after this is done. Just to let you know, we are recording this, and so it'll be available later if you want to watch it again or if you want to share it with friends. A lot of people who can't make it like to follow up. As we're going forward, one thing you can do if you're willing is to go to chat and give us your name and where you're from, just to introduce yourself. You don't have to, but kind of a nice thing to give us a sense of where people are coming from. Now, during the presentation, please feel free to put questions in the Q&A box. I'll be watching those and we'll try to get to as many of those as we can later in the program. But feel free to do that. Just a little bit about Wild Virginia. I can see some of the names. I know some of you are veteran Wild Virginians, but some of you might be new. So just to let you know, we have we have a lot of programs like this through the year, all of which are kind of illustrate the kind of work we do, the things that we care about at Wild Virginia. Our main programs revolve around connectivity and protection of water quality. Connectivity meaning making sure wildlife, whether it's on the land or in the water, can use the areas that it needs to use and do it safely, both for wildlife and for us. That's certainly pertinent to mussels because they need, as you'll hear, fish to be able to move around so that they can survive. We also work a lot to protect water quality, both from horrible projects like Mountain Valley Pipeline and other pipelines, but from all kinds of sources, including toxic chemicals like PFAS. So, keep in touch with us, pay attention to our emails. We give you lots of information through the year and ways that you can be involved to try to make things better. And again, just there are lots of programs like this. We have another one coming up not long from now, having to do with the Appalachian Trail and how it relates to connectivity. So just keep in touch with us. I have to mention that we're right now in the middle of our summer appeal, one of our major fundraising periods. And as much as I don't like to talk about money, I do like to talk about why it's exciting to be involved with us because we do some good stuff. We're a small but I think pretty mighty group that gets a lot done. We don't waste our supporters' money, but I think we do some really good stuff with it. You got a QR code there that you can use, but we're also going to give you some information later at the end of ways you can connect with us. I probably don't even know how to use that QR code, but most of you probably do. We've got 76 people currently tuned in here, which is pretty darn amazing. And that's the kind of thing that we get on a fairly regular basis. So anyway, I want to welcome you. I'm going to stop sharing my screen and introduce you to Alissa, who is our mussel expert. We're thrilled to have her. And take it away, Alissa.
All right. Thanks, Dave. You should be seeing my presentation here.
Yeah.
Just thinking about it. All right, well, this is loading. So I'm Alissa. I am wrapping up my PhD at Virginia Tech in Fish and Wildlife Sciences. And I have been studying mussels for about a decade and a half now. And one of the species I am working with for my dissertation is a Virginia state endangered species called the Tennessee heel splitter. And you'll actually get to see quite a few pictures from my project during this presentation. So the first thing I wanted to do was kind of talk about the marine versus freshwater mussels, because a lot of people, when you say mussels, they immediately jump to marine mussels. There's a few differences, of course, saltwater versus freshwater. That's a pretty standard one. Another one is the foot. So if you've ever eaten marine mussels, which personally I think are delicious, they have a slightly rubbery section. That's their very, very small foot, as you can see here. Freshwater mussels, they actually have a very large foot. It can be up to 80–90% of the mass of the mussel. And if you look at this picture on the right, this white portion right here, that's the foot of this mussel from the Mississippi River. And it's very big and muscular so that those mussels can bury into the sediments and not be washed downstream with currents and kind of hold their place within the river. Another slight difference has to do with byssal threads. Marine mussels, they are not going to be burying into sediments. They actually attach to rocks using these threads called byssal threads that you can see here. And freshwater mussels, as juveniles, as young mussels, do have byssal threads. And this is actually one right here. This mussel is about 2 years old. You can kind of see the clear thread with all the rocks attached to it. Those byssal threads kind of create a drag for the mussel and help it hold its place within the river column and find its place within the sediments without getting washed downstream. And that will disappear as the mussel gets older. That is exclusive to juvenile freshwater mussels. And then the marine mussels have the byssal threads to hold them to the rocks for the remainder of their life. Another difference has to do with reproduction. So marine mussels will just release their gametes, their eggs and their sperms straight into the water column where fertilization will happen, and the veligers will float within the water column until they drop down into the bottom of the ocean and attach to rocks and whatnot. Mussels, freshwater mussels are different. they actually require a fish host to complete their life cycle. Most species are parasitic, and so they need to get their larvae onto the gills or fins of a fish. And we'll talk more about that here in just a second. Okay, so now we will be all freshwater from now on. So worldwide, there's about 1,000 species of freshwater mussels. And North America actually has the most diverse fauna with 300 of those species. Unfortunately, about 70% of that 300 species number are considered threatened, endangered, extinct, or of special concern. So they are not doing well. And we'll talk about reasons for that here in a little bit. If we look at biodiversity in the southeastern United States, we have extreme biodiversity, especially in this corridor here in the southwestern portion of Virginia, moving into the eastern portions of Tennessee and Kentucky and down into Alabama. And this represents high numbers of native fish species, mussel species, crayfish species, etc. In this region. Virginia in particular has about 77 native freshwater mussel species. 54 of those species are native to the upper Tennessee River Basin. So this is again going to be the western portion of the state. And the eastern portion of the state, the Atlantic slope, has 23 species. And of the total species, the total 77 species in Virginia, 39 of them are considered to be in peril. So again, a really high number. Some traits about mussels: they live in the sediments of rivers, lakes, and streams. They are generally long-lived, so you will have mussels that live 30 to 100 years and can even live up to 200 years in some of these really small, cool streams in Washington State, for example. They also have delayed maturity, which means that they don't reproduce for the first time until they are 6 to 12 years old. And this is a big issue because if we don't, we may not see mussels reproducing for years, and we think that there's a healthy population, but there's no new juveniles coming in. So this is a huge factor that contributes to why they are so imperiled. They're relatively large bivalves. That means that they have 2 shells that surround this very muscular foot. And the adults grow very, very slowly and will actually deposit shell material in ring-like structures that we can count like you would age rings on a tree. So that's how we can determine how old the different mussel species are. And while these mussels do have this large muscular foot, they cannot move very far and they cannot move very quickly. As you can see here, this guy is moving. We're not quite sure where he's going, but he is moving. And yeah. And he can move, but again, they are not going anywhere very quickly. So if there is a pollution event or some sort of issue in the river where these mussels are living, they're not going to be able to leave. They're stuck there. And these mussels are filter feeders, which I'll talk more about here in just a second. You may have heard the term native pearly mussels or pearly mussels before, and that has to do with the inside of the shell, which is called nacre or nacra. And it's this pearly, usually white color, but some species, like this is the endangered fluted kidney shell, has a little bit of orange tint. You'll also have ones with pink or purple tint. And that is where you actually get freshwater pearls from. So a pearl is formed because some sort of irritant gets in between the shell and the outermost tissue layer of the mussel, and it's very irritating. And so the mussel will coat it in this nacre to try and relieve that irritation. Just like if you were to get an eyelash in your eye, how your eye kind of coats it to help alleviate that pain, same thing with the mussels. And so that's where you can get freshwater pearls. Now, if you've heard the term cultured freshwater pearls, this is where they actually take a small piece of a mussel shell and stick it into another mussel to create an irritant. And that other mussel will coat it in the nacre and pop out a pearl. And those cultured pearls are actually only about 10% nacre. So they're really only 10% real pearl. The rest of it is usually mussel shell or some sort of other fake irritant. So when you see that, that's what they're referring to. Okay, so I mentioned before that mussels are filter feeders. So they hang out at the bottom of these rivers, lakes, and streams, and they siphon water. And here you've got some mussels. These are rainbow mussels in Copper Creek, which is in southwestern Virginia. And you can see the shells are slightly agape here, and they are actually filter feeding. So that's what they're doing right here. And this filter feeding activity can clean the water. You can do these experiments in real time where you take aquaria and you fill them up with water. Originally, both the aquaria here were full of this kind of cloudy water with lots of material in it. And after 3 hours, the tank that had mussels, which is this one right here, you can see how much clearer it is than the tank that didn't have any mussels. And one mussel can filter 8+ gallons a day, up to 15, even a little more gallons a day for the really large mussels that you would see in like the Mississippi River or the Ohio River. So these mussels can filter an impressive amount of water, and they do so for free, keeping it clean. Some other importance to the filtering of water is the fact that these mussels help move nutrients within the river. So they pull water, or they pull particles from the water column, process them within themselves, and then will release nutrients back into the water or into the sediments via their feces. This is important because those nutrients going into the sediments can help stimulate primary producers like aquatic plants, which can provide habitat for young fish. They can also help stimulate populations of benthic invertebrates like dragonfly and caddisfly larvae, which are aquatic during that portion of their life stage. And those of you that are avid fishers know that those aquatic invertebrates, dragonfly and caddisfly larvae, as fish food. So indirectly, by providing these nutrients to primary producers and other aquatic invertebrates, mussels can help stimulate fish populations. So why are mussels threatened? There's a whole host of reasons, and we're just going to briefly talk about some of them right now. One you may have heard of, especially if you've ever been to Iowa or anywhere along the Mississippi or Ohio River, and that is the pearl button industry. Back in the late 1800s, late 1880s, John Bopel, who was a craftsman who made buttons out of like hooves and antlers, he thought freshwater mussel shells would be a great way to make buttons. And so he opened up a facility in Muscatine, Iowa, also known as Pearl City, and you can see their building down here, and started producing these pearl buttons. By 1898, they were producing over 130 million buttons per year at that facility. Unfortunately, how they collected the mussels led to a decimation of mussel populations. They used these hooks that are called braille hooks that you can see over here on the left. And they lined them up on this pole and would drop that pole with all the hooks into the river and drag it along the bottom. And mussels, when they're filtering, will be open. Their shells will be slightly agape. And when something like this hook comes into contact with the inside of the mussel, it will instinctively shut. And so the mussels would all shut on these hooks, and they would be able to pull up hundreds of mussels at a time out of the river. And to make matters worse, there were only certain mussel species that were actually able to be used for buttons. It depended on the shell shape and size. If the shell was too bumpy or if it had ridges, which some species do, they wouldn't be able to use it. So the ones that were used for pearl buttons would go to be made into pearl buttons, and the ones that couldn't be used for buttons would actually go to feed the hogs. So no mussels were going back to the river in this situation, and it decimated populations in the Mississippi River. And when that happened, they picked up operations and moved to Arkansas, Ohio, and Tennessee. So further contributing to decline in mussel populations. This one's really cool. This is one of the shells that they punch pearl buttons out of. And sometimes you can find these at antique stores, especially in Iowa. And so you can see all of the punches that they did to create the buttons. And here's some of the buttons. This is a yellow sand shell mussel. Other factors contributing to mussel population declines include pollution. So this is entering chemical spills or trash into the river system that can affect the mussels. Remember, they can't move very quickly or very far. So if pollution enters the stream or the river where they are, they're going to get hit with it. We also have invasive species. This is another one that thankfully we don't have to deal with in Virginia. But if you've ever been to the Great Lakes, the Mississippi River, etc., you have probably seen signs and postings indicating how important it is to clean your equipment, because these guys can travel very, very easily. And when they enter a water body, they spread rapidly, they reproduce rapidly, and they will attach to anything and everything, including this live mussel. This right here was a live mussel from the Mississippi River that was buried to about right here in the sediments. And the zebra mussels attached to the shell, preventing it from filter feeding, so preventing it from getting food and oxygen, and will ultimately lead to its death. So they are terrible for mussel populations and actually rivers in general. Other threats include urbanization and deforestation. So urbanization, like adding more blacktop, more asphalt and parking lots, and deforestation, removing trees and shade. Both of these can contribute to warmer water. Obviously, removing the trees can cause more sunlight to reach the river, increasing the water temperature. And more blacktop will actually cause issues with runoff. So if, we've had a very hot summer here in Virginia. I'm sure many of you remember that very well. It's cooling down a little bit, but it was very hot for a while there. How hot that blacktop gets underneath the sun, okay? So it just heats up. And then when there is a rain event, the rain hits the blacktop, and as it's moving to the stream, as it's prone to do, it will pick up heat from the blacktop, and the water that goes into the river or the stream will actually be warmer than the water that originally hit the ground. So again, we're leading to increases in water temperature. We can also have road salt that gets into the river systems. Here in Virginia, we salt our roads when there's going to be wintry weather, and that road salt goes into the river system when it rains or snows and the ice and the snow melts. And road salt can be toxic to mussels, especially the juvenile stages, and can lead to issues there. And then a big one is climate change. And a lot of my research focuses on how increasing water temperatures from climate change can lead to issues with mussels. They are what we call ectotherms. They are very dependent on the river temperature to determine their heart rate, their growth, their general physiology. And when we increase those water temperatures or change those water temperatures, it can lead to stress. And just like in humans, when you become stressed, your physiology, your biology can start to be affected. We see that with mussels as well. Now, other issues with climate change include altered precipitation patterns. So years of drought, years of extreme flooding. We've had very horrible hurricane years with extreme flooding. I believe 2018 was a really bad year. That is in recent memory. So all of this can contribute to mussel populations being threatened. Also, dams and river impoundments. So we have some, like here, this is in, I believe, the Mississippi River. This is going to be a big river thing, these lock and dam systems. But we also have them in smaller rivers here. Virginia's actually been working to remove a lot of these smaller, obsolete dams that are not necessary anymore, to help bring the river systems back to their natural state. These dams create cool, deep sections immediately downstream, and they disrupt the natural flow of the river, and they can cause sediments to drop out of suspension and bury mussels, and just create a river system that's not hospitable to mussels. And as we talked about before, mussels are parasitic for part of their life cycle, and these dams can actually prevent fish migration, which means that they will not be able to reproduce. And it can lead to, again, population declines. So the life cycle of freshwater mussels is one of the coolest things ever. This is why I fell in love with mussels. Again, most species, not all, but most species are obligate parasites on the gills or fins of a fish for part of their life cycle. So with freshwater mussels, the adult mussels, so that's down here in this illustration, the male mussels will release their gametes into the water and they'll float downstream. And when she's filtering, the female mussel will pull them in and fertilize her larvae or her eggs. And then she holds the larvae, which we call Glochidia, until the time that it's needed to get on the fish. And the way that she does that is incredibly interesting. We're going to talk a lot about that here in a second. Then these mussels get a free ride around the river system. Remember, adults are not going to move very far. They can't. So in order to persist throughout the river system, being on this host fish allows them to get a ride up and downstream and help persist in those upper portions of the stream. Whereas if it was just adults, they would constantly move downstream with the river current. As they get their ride in the fish, the mussels transform into juvenile mussels, which will then drop off when they are ready and bury into the sediments and start this cycle all over again. So we'll look at the glochidia first, again, the larvae of these freshwater mussels. Here is actually a picture from my project. These are Tennessee heel splitter mussels. Again, this is endangered in the state of Virginia. Tennessee heel splitter, this is the adult mussel. Somebody was very generous with the heel splitter name, but they're still really cute. So these guys kind of look like Pac-Man. These glochidia actually look like little Pac-Man, and they'll be open when they're released from the mussel. And they've got these two shells with a hinge here. And when they come in contact with a fish, they will snap shut and then insist into the fish's tissue. So again, they have a hinge structure. This is a closed glochidia here, and here is the hinge. This is an open one with the hinge here. And some species have hooks. This is not all species. The glochidia size and structure is actually different between species of mussels, and I'll show you more examples here in a second. But this particular species has these very large hooks that you can see here to help it attach to the fish. Here we've got an example of two different sizes, some very, very large glochidia. These are without hooks compared to some of the very small glochidia. And again, this is different between species and is a distinctive characteristic used to identify them. And then here are some glochidia encysted on the gills of a host fish. And I've got a few more images of that coming up too. So again, we're back to my project. This is the Tennessee heel splitter. It's a little tricky to see, but I want to show how big these glochidia are. This right here is a drop of water, and you can kind of see there's little red, brown spots in there. Those are glochidia. And this is a picture that we took with a camera phone. So this is not like any microscope, any fancy camera. This was a camera phone. And so all of those glochidia in that little water drop will eventually try and get on a fish. And this particular mussel species attaches to the fins of fish. And so in this red circle, you can kind of see, I apologize if it's a little difficult to see, but there's little red bubbles here. Those are the glochidia attached to the fins of that fish. Again, pictures taken with a camera phone. There is no microscope. You can see these with the naked eye. For smaller species, like this one is the snuff box mussel, which we will also talk about in a little bit. You can see all of these little bubble-like structures that are attached to the gills of this fish, which is a log perch. Now we're going to bounce to the juvenile mussels here. Again, they pretty much just get a free ride upstream on their host fish, and they drop off once they've developed into juveniles. And they are very small when they do first drop off. Very, very tiny, see-through, but they are active. Their feet are moving, they're feeding, they're moving around trying to find a decent place to start developing. And we do sometimes see differences between species at this stage. So these are newly released juveniles. These mussels here are from various projects I've been a part of. They are 2 to 4 months old here. So you can see these two are mucket species. They're very similar. They're very oval shaped with some smooth shells. This is the washboard species, and this species will actually grow to be huge. They live in like the Mississippi River. They'll get to be about the size of your head, like so big that Native Americans used to use these shells for gardening tools. And you can see that they are different from the mucket species. They have these slight indents in the shells here. And then this is that Tennessee heel splitter mussel again, and they actually have little ridges on the outside of the shell. So there are distinctive characteristics that we can see at the juvenile stage as well. Now, those small juveniles we are not going to be finding in the field. We usually won't see them in the field until they're about a year old. This juvenile is getting close to a year old here that we collected, I believe, out of, again, Copper Creek in southwestern Virginia. And you can see this little rock here. It's still attached to its byssal thread. It's got a very tiny byssal thread, and it's a very small mussel. These mussels are about 2 years old out of the upper Mississippi River. And you can see they're about the size of a quarter there. So now the coolest part of freshwater mussel life cycle is how this mussel gets her larvae onto a host fish. So there are actually different ways, different categories that we put reproductive modes for mussels. We've got what's called a broadcast spawner, a conglutinant producer, a mantle modifier, and an aggressive host trapper. And I'm going to go over each of these here in a second, but I do want to point out that these reproductive modes are linked to how specific a mussel is for its host fish. Some mussel species are able to get their glochidia onto many different host fish. Others are can only use one or two. And this is not just an access thing. This is glochidia will not transform into juveniles on the host fish if it's not the appropriate host fish. So we see host generalists able to use many, many different fish as host species, and host specialists like aggressive host trappers, for example, will tend to just be able to use one or two fish species. So a broadcast spawner is a mussel species that will release her glochidia straight into the water column. They kind of float in the water column and will encounter a fish largely by chance. These species can usually successfully transform on a wide range of host fish species, and they are also pretty ubiquitous in their range. So the three-ridge mussel here, this is a very ubiquitous mussel, very common in the Mississippi River, and its range goes all the way down into Tennessee. The eastern elliptio is also very common, especially on the east coast of Virginia and North Carolina. This is one of those Atlantic slope species, one of the most common species in this region, or in that region, is this mussel right here. Now, conglutinant producers actually create this packet of glochidia that's called a conglutinant, and they have modified it to resemble fish or invertebrate larvae or worms or something that looks like food. So these mussels are trying to attract a predator fish by offering it something that looks like a free, easy meal. Okay. So you can have ones like this. This is, I believe, a fan shell mussel whose conglutinants look like little worms here. Some of them will be even colored. Here's another fan shell example. These are red, and you can see they're about the size of a dime. And then the dromedary mussels here, again, red coloration and kind of look almost leech-like. And again, this is an attempt to attract a predator fish with what it thinks is a free meal. You also have species like the kidney shell here that modifies their conglutinants to look like little fish. So on the left, this is a real fish. And then these are the little conglutinants that are kind of just, you know, floating just at the surface of the rocks, maybe just grazing the surface of the rocks. And it looks like something that's a really easy prey item. And I apologize if you can't see it. These are very, very clear here. But when the mussel, or when the fish goes in to eat this conglutinant, it ends up getting a mouthful of larvae, as you can see here in this open conglutinant, instead of a food item. And this is that same mussel, that kidney shell mussel. These rows here that look like pearls, those are the conglutinants waiting to be released from the mussel. And we actually collected this mussel in southwestern Virginia here. A similar species to the kidney shell that you just saw is the fluted kidney shell. This is the endangered one that I mentioned with the orange nacre earlier in the presentation. This mussel species actually modifies its glochidia to look like black fly larvae. And you can see here we've got the fish with the black fly larva conglutinates that look like really easy food items. And again, just packets full of Glochidia that will go right into the fish's mouth. The next one is the mantle modifiers. And the mantle is the outermost portion of tissue that touches right up against the shell of the freshwater mussel. And these mussel species will actually modify that tissue to resemble a small fish or a crayfish, or they'll inflate the mantle like this species is doing right here. And again, that's designed to look like a prey item. So these will even move. They can move these tissues so it looks like a little small swimming fish. And sometimes they'll have eye spots. This one is designed to look like a crayfish here. And you can see these little pouches, these white pouches, those are actually full of Glochidia. Again, we're trying to attract a predator fish like a largemouth bass that'll come in, bite that mantle lure and get a mouthful of Glochidia instead. Just a few more examples. You can see they've got these very intricate tail-like structures, some eye spots. And again, here, that tail-like structure, there's the eye spots. And these little ridges here are all full of Glochidia. So when the bass or the other predator fish comes along, thinks it's getting a really easy meal because, you know, this fish isn't going anywhere, it comes up, bites it, and instead gets a mouthful of Glochidia, which is this cloud right here. So pretty fancy. And I do have a video, hopefully this plays here. It does go a little slow, but you can see this is a pocketbook mussel lure. Yeah, I apologize that it goes a little bit slow, but you can see the kind of pulsing of the fish like lure and the eye spots. And I have been doing this for a decade and a half, and I have to tell you, I have been fooled before walking along the sediment or along the riverbank, and you see something like this out in the water, and it's actually a mussel and not a fish. So it's pretty darn cool. And one more example here, this is the modified mantle tissue, and these are the actual fish. So again, amazing for something that doesn't have any eyes to be able to create such an impressive mimic. Now, my personal favorite, the aggressive host trappers. So these mussels actually lay and wait for their host fish to come along. And so an example here is the very appropriately named snuffbox mussel sits within the rocks with her shells slightly open, and you can see this is her inner portion here, and a fish, in this case, they use a log perch as their host fish, which rolls stones and stuff as part of its general biology. And that log perch will come along and will roll right into the mussel, and then the mussel will snap her shells shut and actually push Glochidia straight into the mouth of the fish. And they have these teeth-like structures on their shell. These are called denticles, so like dentist teeth here that they use to help hold that fish. Unfortunately, a lot of these species, if not all of them, I believe now, are endangered. Again, because they really only use one or two host fish for their reproduction. And here you can see the log perch within the shells of the snuffbox mussel here. And I do have a video, hoping it plays here, maybe. Thinking about it. Okay. All right, so you can see this mussel, the snuffbox mussel, here, her shells are open. You'll see her move here in just a second. And then you'll see a slightly anesthetized log perch come in right here. He goes to flip the rock and snap. And she will actually hold that fish until he stops struggling, until the fish stops struggling. And then what she'll do, and you're going to see this on the next video, is she will slightly open her shells, allowing that fish to take a big breath and push her Glochidia right into its mouth. So it's very effective. All right, so let's see here. Again, I apologize. I was having some issues with the videos earlier, so hopefully this next one plays as well. We're thinking about it. But again, all right, here we go. And we'll watch this one twice so you can see what goes on here. So again, she's going to hold that fish until the fish stops struggling. And then the first thing that is going to happen when that fish takes a breath is going to get a big mouthful of Glochidia. All right. Maybe we're going to play. All right. We're thinking about it. Anyway, snuff box mussel, while this loads, it is in Virginia. It's native to Virginia, but its range actually goes all the way up into Minnesota and Wisconsin. So this species has a pretty impressive range. It is listed as endangered in the U.S. endangered species list, as is the other aggressive host trapper that we'll talk about right there once we go back and hopefully watch this video. I do apologize. They're fairly large files. I'm so close. I only had 3 and it's the last one here. Here, let me try getting out here. Oh no. All right, well, we'll see if that happens. Okay. If not, we'll watch this at the end. It's a really cool video. All right, it doesn't look like it's going to go, but we'll watch it at the end here because it's really awesome. If I can get it going. I seem to be stuck a little bit here. All right, so while Microsoft here, all right, is thinking about what's going on, this is the oyster mussel, and this is another aggressive host trapper. And this mussel has mantle tissue that she inflates. And it's actually a really white color, or like even sky blue in some individuals. And so she'll sit at the bottom of the stream with this very clear open mantle tissue that even has, as we'll see on hopefully the next slide, a little micro lure that moves and attracts those host fish. And when the host fish swims in between the shells, she will actually snap her shells shut and inflate that tissue and hold on to the fish, kind of cutting off its oxygen supply. And once the fish calms down, okay, so you can see here she's got her shells snapped shut, and that is the tail of a fish that's in between her shells. Once the fish calms down, she will deflate the mantle tissue and she'll actually push her Glochidia straight into the mouth of that fish and then release the fish to go on its merry way. Now, I've told you about how awesome mussels are, and I've also told you some pretty tragic things about how decimated these populations are, but I do want to let you know just briefly how we're managing these mussel populations. And a lot of that has to do with propagation. So we actually do this portion from the Glochidia to the juveniles in the lab. This is a very delicate relationship in the field. If the fish were to be harmed, or for some reason we're not in the area where the mussels were, you can have a missed opportunity for reproduction that year. Fish can get killed with pollution events, and you already have pretty low survival of juveniles in the wild. So we try to mitigate that by doing this in the lab. So we'll actually go out and collect gravid or pregnant female mussels from the river, and we will check to see that they're gravid, which we can either use our fingers to just slowly pry open the shells, or we can use, which is what Tim is doing over here, or we can use things, this is actually a nasal speculum that we slowly open their shells and check to see if she's gravid. This is a plain pocketbook mussel that I was checking here, and you can see these little, just barely see this area here indicating she is gravid. And so we will bring these mussels back to the lab. We will extract the Glochidia and we will put it on fish. We will infest fish with this Glochidia. Then we will house the fish, collect the juveniles, and raise the juveniles until they are old enough to go back out into the stream, which is about 1 to 2 years, depending on the species and the time of the year. So again, this is from my project with the Tennessee heel splitter. I'm using a hypodermic needle to kind of open up the tissue where those Glochidia are kept. And again, I apologize if you can't really see it, but all of this red here, those are the Glochidia from this species. And then we've seen this picture before where they all look like Pac-Man. And so we take this Petri dish full of Glochidia and we put it in a bucket with some water and some fish, and a bunch of air stones that help keep the Glochidia moving and make sure that the fish, it has enough oxygen and isn't too stressed. And as you can see, this guy is clearly having a great day in his little bucket and getting infested with Glochidia. And then we check to see if there was successful infestation, which again, we've seen this picture of my Glochidia attached to the fins of this sculpin. Then we house the juveniles in aquaria in our lab at a set temperature. We keep them as stress-free as possible. And in 10 to 14 days, juveniles will start dropping off of those fish and we can siphon the bottom of the tank and pull out the juvenile mussels. And we put them into systems, like this, which each of these green tubes here is going to a different box with some sediment and juveniles in it. And we will grow them out 1 to 2 years old. This is just one example. I want to point out there are many different systems. So if you've heard of other systems, there are a bunch of different ones depending on the facility. I just briefly wanted to mention these here. And then when they are old enough, we will bring them back out to the river. And again, about 1 to 2 years old, and we will put them back out. We do mark them with tags. Like here, you've got green colored tags. Sometimes we do laser tags. There's a facility out in Charles City, Virginia, has a laser engraver, so you can laser engrave your tag on them. And that way we can track them during surveys throughout the coming years and see if they are growing, if they are still alive, and if they are reproducing. And with that, I do want to give a big thank you, first of all, to all of you for listening to this presentation and for joining, and obviously Dave in Wild Virginia for hosting this. Photos came from my advisor, Dr. Jess Jones. Also, Zach, Isabel, Anna, Katie, and Tiffany are all previous colleagues at Virginia Tech. And then Chris Barnhart was responsible for a lot of the videos and the images here. And he, I believe, is retired from Missouri State, but we would not know anywhere near as much as we do about mussels without him. And before I take questions, I'm going to try and, or while we take questions, I'm going to try and get this video to play because it's so cool. And maybe it'll work now.
Okay, while we're waiting to see if this works, let me at least work through some of the questions. There's some good stuff here. What is the best, and if it starts, tell me to shut up. What is the best way to identify and locate mussels in small streams in Virginia? What's the, oh, there it goes. There it goes.
Yeah. Okay. So you'll see here little dots all around this fish. Those are the Glochidia getting pumped directly into the fish's mouth. I can play it one more time, maybe. And so she'll open her shells and push the Glochidia straight into the fish. There we go.
Amazing.
Yeah. All right,
Okay.
I can stop sharing.
Cool. So again, what's the best way to identify and locate mussels in small streams in Virginia? What's the best tracking method?
So for us, so mussels tend to aggregate into assemblages that we call mussel beds. And so in a given river, they're not located throughout the entire river. They'll just be located in a small section of that river, and there'll be different patches of these mussel beds. And when we're looking to locate mussel beds, we look for things like riffles. Mussels really like riffles. It's good oxygen flow, usually good food coming in, stagnant water. Sometimes you will find mussels there. I mean, they're species that can thrive in places I would not have expected. But usually riffles with good water flow, decent shade cover. Those are areas where we would find mussels. And then when we track them, we do keep at least at the facilities in Virginia, we keep logs of where we go and when we go. And we do try to follow up to see if those populations are still persisting over time, especially for endangered species or threatened species. And so those ones, it's usually the state individuals that know that information, but we will tag all the mussels that come from our facilities, as well as sometimes, for example, for my project, I tagged every mussel that I touched for my project, because part of my work with the Tennessee heel splitter was to figure out if populations were persisting over time. So I need to know if I'm encountering the same mussel over and over again. So most of the people in Virginia know that if they see a blue L tag, it's my tag from my mussels, and they can say, hey, Alyssa, when's the last time you saw this mussel?
Cool.
Yeah.
How many freshwater mussels are edible?
This is a great question. Technically they are edible, but big but. First of all, many of them are endangered. It is illegal to obtain an endangered mussel. It is also illegal in Virginia to collect any mussels. So from the legality side, we don't want to do that. But technically they are edible. I would not recommend it. First of all, I mentioned this way back at the beginning with marine versus freshwater. The marine mussels will have that foot, and sometimes if you're eating them, you can, it's pretty rubbery, but it's small. But the freshwater mussels is gonna be like 80 to 90% of that rubbery hard material. It is not gonna taste good. It's not very nutritious. And there's a lot of pollution in our freshwater systems. So it's probably a good way to glow in the dark, consuming a lot of them.
The
So I
meal
wouldn't
either with it.
but technically, we do know that Native Americans did include mussels in part of their diet, especially during parts of the year when other food sources were less available.
Okay, here's another one. Dams, if dams are detrimental to the mussels because of the fish movement, what about beaver dams?
That's a great question. So I actually had a site with a beaver dam. I was at this site probably twice a year over 3 years, and about halfway through, a beaver moved in. Now, the beaver dam, I do still think there was fish able to move through that particular beaver dam. It would have to be a pretty big one to block off the whole portion of the river. But the main issue that the beaver dam caused was it really blocked up the water channel, and it ended up being incredibly deep. It went from being about my knee height, I'm 5'7", it went from being about my knee height water level to up about mid stomach, chest area. And actually one of the other individuals on here, he was out with me trying to get mussels, and we kept flooding our waders because the water had gotten so deep. And with that, it got really cold, which is also potentially damaging for mussels. But that's a great question. Of course, if it was a big beaver dam, I'm sure it could cause issues, but at least they tend to be a little bit more natural and can also be removed with like flooding and stuff like that.
Okay, hold on, it shifted on me. While attached to the host fish, are mussels getting nutrients from the fish? And do the attached mussels harm the host fish?
That's another excellent question. So the characteristic of any good parasite is that you don't want to kill your host. And there's been a lot of research into this, and it looks like for the most part, the mussel is just getting a free ride upstream. There is some research that looked at the transfer of carbon between the mussel and the fish, and there was some transfer indicating some tissue or some ion movement between those between the mussel and the fish. But for the most part, it seems to be just a free ride upstream. And the fish usually survives unless it's a very heavy infestation, which we have seen in the lab where we accidentally overinfest and it blocks up the gills and the fish can't breathe anymore. So yeah, but usually they survive.
Okay, here's one about using gloves, or how you use, or what precautions you might take to keep from transferring bacteria or other things when you're moving the mussels into the stream or other activities.
Again, excellent question. And also very relevant because some of you may have seen an article that came out last summer, maybe the summer before, about potential pathogens in the Clinch River in Tennessee. that can transfer to mussels and what might be causing some mussels to die. We are very careful to not mix mussels from different populations or from different streams in the lab. For example, we keep all of our Tennessee River system mussels in one building and all of our Atlantic Slope mussels that we are propagating in another. And we track where the mothers come from, and we track which females we're pulling in each year. Those mussels always get a tag, so we know who is using which mussels for propagation, so they're not used too frequently. And you know, we do make sure that our facility water, you know, we're not changing water and whatnot. We try to keep that to a minimum. You know, we're not bringing water from the field and dumping it in our tanks, but we do have pond water at Virginia Tech that we can monitor and make sure is not detrimental to the mussels. Great questions.
There's a bunch more, and I will say that we have 5 minutes left, and I don't think we're going to get through them all. But what I try to do is make sure to save these, and maybe we always follow up with folks, whether you attended or all the registrants will get a follow-up email, and maybe we'll see if we can answer some more of them in our follow-up, because I don't want to leave you hanging. But just maybe one or two more here, and then I'll have a closing message for you. With turbid and oxygen deficient water, such as that released periodically from high head dams, impact mussel populations? I think I know the answer to that.
Yes. Yes, they can. So mussels have a set amount of oxygen requirements. They are capable of withstanding periods of low oxygen because they can shut their shells. And so they can withstand that for a certain amount of time, but not for incredibly long periods. Same with turbid water. They can shut their shells and kind of ride it out for a little while, that excess sediment and whatnot. But eventually they will have to open their shells to filter feed and remove waste and whatnot. And yeah, then it gets dire.
Okay. And there's something here about controls for zebra mussels and others like that. And while are there biocontrols for zebra or quagga mussels? And while you answer that, I'm going to start sharing my screen a little bit to give us our final slides.
Yes, so zebra mussels, the main issue is that their larvae do not require a host fish, and they are actually capable of withstanding pretty high temperatures and desiccation, so no water for a long period of time. So if you are in any areas with zebra mussels in them, you have to really wash your equipment down and let it dry out in the sun. I believe it's 2 days minimum to kill off those larvae and prevent it from moving to a new location. And obviously states like Minnesota and Wisconsin have intense fines and protocols in place to make sure that fishing equipment, minnow buckets, for example, when people go fishing with their minnow bucket, pulling water from one location and then dumping it into another is really bad because those little larvae can be in there and get into a new system. In Virginia, we do have protocols to clean our equipment and make sure that we're not moving pathogens between different river systems here. But again, we do not have zebra mussels in Virginia. Hopefully it stays that way.
Okay, well, again, I'm sorry we didn't get to every single question. We'll try to follow up as best we can. Thank you so much, Alissa. To me, this is a fascinating topic, and I can tell you that the fact that 70 or 80 people stuck around to the very end tells us that it's fascinating to a lot of other folks. I'll just end by, again, making a pitch for Wild Virginia. We do these kinds of projects or these kinds of programs, and what we're doing out there in the world is to try to make conditions so these kinds of species and the many others that we depend on can survive. We do have yet another week in our fundraiser for the summer. Anything that folks contribute is very useful to us. We depend on hundreds and hundreds of people who are really great supporters through the years. And, so we value that. One thing I wanted to mention, and we don't have much time, I told Alissa I wanted to mention something that she's part of. It's
Yep.
called the Virginia Scientist Community Interface, or VSci. And we'll send you a link to that also. It's a group of people like her, graduate students who help in figuring out some of this great stuff, but also feed it into advocacy efforts. And they've been really helpful to us and other groups. So with that, I'm going to sign off. Thank you all again for being here. Look for our follow-up email and look for our other shows coming up. So thanks, everybody. Okay.